Good morning, everyone, and welcome to today's webinar with Dr. Kevin Shoulars, part of our Experts in Hematopoiesis series. My name is Amanda Fentman. I'm a scientific marketing specialist at STEMCELL Technologies, and I will be your moderator today. I'm pleased to welcome Dr. Shoulars, who will be giving his presentation entitled Development and Validation of a Rapid ALDHbr-Based Cord Blood Potency Assay. Dr. Shoulars currently serves as supervisor of the Duke Potency Laboratory at Duke University Medical Center. The Potency Laboratory is part of the Carolinas Cord Blood Bank under the directorship of Dr. Joanne Kurtzberg. Dr. Shoulars is responsible for the development and deployment of procedures for the measurement of potency in umbilical cord blood units intended for transplantation. He completed his doctorate in cellular and molecular biology from Baylor College of Medicine in 2006 and joined the Carolinas Cord Blood Bank at Duke in 2008. And now, without further ado, I will hand it over to Dr. Shoulars.
As Amanda said, my name is Kevin Shoulars. I'm a senior researcher at the Duke Translational Research Institute at the Carolinas Cord Blood Bank for Duke University Medical Center. Today I will be speaking to you about the development and validation of a rapid aldehyde dehydrogenase-based potency assay for measuring cord blood units. Up to 10 to 20% of cord blood units selected for transplantation fail to engraft. We hypothesize that the reason for this to occur is due to decreased potency of the cord blood unit graft, and that measuring the cord blood unit potency could improve cord blood selection and also improve outcomes after transplantation. In this context, potency is the ability of a cord blood unit to engraft following transplantation. Therefore, a potency assay is a measurement of one or more parameters of the cord blood unit that predicts the efficacy of the unit following transplantation. Currently, several parameters are used by transplant centers to select units. These include the total nucleated cell count and the human leukocyte antigen matching measured on the cryopreserved units before cryopreservation. In addition, various labs examine pre-cryo colony-forming units and pre-cryo CD34 cell counts with or without viability. Despite these current parameters used in the cord blood unit selection, cords continue to fail to engraft, possibly due to the poor quality of units. Therefore, better assays are needed. Previous work in laboratories at Duke have shown that the best current potency assay to predict engraftment after cord blood transplantation is the post-thaw CFU measured on a unit when it is thawed for transplant. Here, looking at 159 patients from historical data published by Prasad et al. In 2008, transplants are divided into two groups: those where the patient receives more than 56,000 CFUs infused per kilogram, and those receiving less than 56,000 CFUs per kilogram. As shown, the overall survival was superior in the group receiving higher CFUs. Though this assay is useful, it is not the best for using as prospective assay due to the time required, which can be up to 14 days, and the variability between operators. Several current alternative methods are under investigation, including the HALO assay, shorter colony-forming unit assays, and automated colony-forming unit assays. In my lab, however, we are exploring the usefulness of aldehyde dehydrogenase as a potency measurement. Aldehyde dehydrogenase is an enzyme that oxidizes aldehydes to carboxylic acids. It has been shown to be enriched in hematopoietic stem of progenitor cells. Cells which express a high level of this enzyme are clinically relevant for transplantation, and importantly, much like the CFU assay, cells detected ALDHbr assay must be viable to score positive in this assay. To give you a point of reference, a cord blood unit is preserved in a bag consisting of several different compartments. The majority of the unit is stored in two compartments comprising 80 and 20% fractions. These fractions can be combined as a single infusion or used as separate infusions during transplantation. In addition, the tubing that is used to fill the bag is divided into several segments, seen here, which prior to cryopreservation and which are kept stored with the bags to be used for things like the HLA assay. These segments can be removed from the unit for testing prior to release of the transplant center without compromising the integrity of the larger 80 and 20% fractions. For identification of cells rich in ALDHbr-Based, the AutoCount reagent was developed at Duke with refinement of the assay at Aldergen in Durham, North Carolina. Briefly, the aminoacetaldehyde with an attached fluorochrome called BODIPI and an inhibitor was created. The reagent is activated by adding acid to remove the inhibitor. This compound can then freely move in and out of cells. In cells that are rich in the aldehyde dehydrogenase enzyme can then convert the aminoacetaldehyde into aminocetoacetate, which is retained by the cells, causing them to fluoresce. These cells can then be detected by a cytometer, as seen on the right in this gate here, which is easily separated from the majority of the non-ALDH-Bright cells. The addition of a inhibitor called diethylbenzaldehyde inhibits the reaction so that the cells are not detected within the gate. The primary problem we found with converting this assay for use on the segments shown earlier was that during the assaying of fresh cord blood units, which the assay was originally designed for, red blood cells could be removed with lysis. However, the lysis of segments causes a large amount of debris and reduces the ALDH-Bright and CFU content detected. Therefore, in assaying the segments, a glycophorin A assay could be used to detect these RBCs. This allows the large amount of RBCs present, seen here in pink, and in the graph here, to be excluded during analysis to give a better determination of the actual ALDH-Brights present, as seen here. To give an example how this is effective, several fresh cord blood units were tested prior to cryopreservation. Because these units were lysed, the units with and without GlyA had similar percentages of ALDH-Bright, seen here as 1.14 versus 1.21, 1.39 versus 1.46, and 0.09 versus 0.18. However, when measuring the units on the segments, because of the large amount of red blood cells, each of the segments initially without the GlyA has a low amount of ALDH-Bright compared to what would be predicted based on the fresh cord. However, once the GlyA was added, you can see that the ALDH-Bright percentages were roughly equal to what they should have been in the fresh cord blood unit. The initial version of the assay was performed by thawing a cord blood unit segment and removing the needle with the blood with a needle. The remaining blood was diluted with dextroalbumin, whereupon 100,000 white blood cells were removed for the CFU assay. Cells were washed with HSA PBS and incubated with the activated ALDHbr reagent at 37 degrees. Following this incubation, an incubation with phenotyping antibodies, CD45 and CD34, along with a separate tube for the glycophorin A for determining the red blood cell contamination. After a final spin, the 7-AAD viability marker could be added and the flow cytometry analysis could be performed. This is done by gating the cellular populations shown here. A forward and side scatter gate was created to eliminate debris. We then gate on CD45 positive cells, non-viable 7-AAD stained cells, and CD34 positive cells, and finally on ALDH-Bright cells shown here in red. These gates tell us the relative amount of the various cell types within the segment. Excluding the non-viable cells gives us the viable cells in each population. After this assay was performed, a retrospective study to test the hypothesis was done on segments from 38 cord blood units previously transplanted at Duke with available segments from patients who had either rapidly engrafted or never engrafted. 21 rapid engrafters, here defined as transplants reaching an ANC of 500 in less than 20 days, were selected along with 17 non-engrafters. The segments were then tested and the pre- and post-thaw populations were calculated. Several of the more traditional parameters were important, however, they were not the most significant. The total ALDH-Bright times 10 to the 5th infused per kilogram based on the segment data was the best predictor of engraftment. As seen in the chart on the right, where rapid engrafters depicted them as the blue dots and the non-engrafters here depicted as red dots were plotted. based on the days to ANC 500 versus the ALDH-Bright infused per kilogram. Non-engrafters here were assigned days over 100 for graphing purposes. Here you can see a clear distinction between the rapid engrafters and the non-engrafters, with a threshold of approximately 40,000 ALDH-Bright cells infused per kilogram. You will note that this is similar to what we showed earlier with the CFU assay and post-thaw transplanted units. Following the retrospective study, the assay was changed to allow for the testing of segments from cords requested for confirmatory typing. Whenever a request comes in, the cord is identified, a segment is pulled from that cord, and normally was sent for HLA typing by itself. However, as the segments are a precious resource, we modified the assay so that the blood from the segment could be spotted onto an FDA card for the HLA typing. The sample could then be serially diluted with the step of dextranalbumin dilutions, resulting in a eventual tenfold dilution of the segment. An aliquot from that was taken for counting on a Sysmex to determine the cellular content. 100,000 cells from this aliquot were removed for plating in Methicult and counted 14 days later as part of the CFU assay. The original sample was then spun down in a series of centrifugations and wash steps to rid the sample of the dextranalbumin, during which time the out-of-count reagent could be prepared. Briefly, the out-of-count reagent is activated with an activator, which consists of hydrochloric acid, and a 20-minute incubation at room temperature. There's then a neutralization buffer to bring it back to a neutral temperature and stop the activation. The sample from the wash step can then be added to the out-of-count reagent and followed by a 30-minute incubation at 37 degrees. Following this activation, the samples are placed on ice and a prepared antibody tube is added. The antibodies here consist of the CD34 and the CD45. However, we've changed the original version of the assay so that we added different GlyA antibodies so that it could be detected in the same channel as the 7-AD, enabling us to use a single tube to determine the phenotype. The samples could then be acquired on the cytometer. We were able to set an acquisition target of 100,000 viable CD45 cells for each segment just to standardize between segments. The flow analysis with this method looks very similar to what we saw earlier, where forward and side scatter gauge is used for debris. In this case, the 7-AD, which is roughly in this area of plot B, and the GlyA come in the same gate. The gating of the CD45 and the CD34 and the ALDH-Bright can then be done as before. However, it is easier to do now that the red blood cells are removed. And finally, we have the inhibitor, which shows that the ALDH-Bright gate is indeed inhibited. As the Carolinas Cord Blood Bank has a biological license agreement through the FDA, we had to develop a validation plan for the ALDH-Bright assay. In the future, all validated cord blood units are going to require a validated potency assay for release from the bank under the BLA. This was written according to guidelines set by the International Conference on Harmonization, the Harmonized Tripartite Guidelines Q2R1, in consultation with regulatory authorities. Briefly, we tested 5 parameters from the guidelines: accuracy, repeatability, linearity, intermediate precision, and specificity. For accuracy, defined as the closeness of agreement between the value which is accepted, either as a conventional true value or as an accepted reference value, and the value found. The samples tested for ALDH-Bright for this part of the assay were required to be within 20% of the expected value. We then tested repeatability, defined as precision under the same operating conditions over a short interval of time, where tested samples were required to have less than 20% variability within each sample concentration. Linearity, defined as the ability within a given range to obtain test results which are directly proportional to the concentration or amount of the analyte in the sample. which required the correlation between the target values and the tested values in our assay. Intermediate precision, which is the within laboratory variations, different days, different analysis, different equipment, etc., require the testing variability between the operator, sample, and instrument to have a CV of less than 20%. And finally, specificity is defined as the ability to assay unequivocally the analyte in the presence of components which might be expected to be present. This required the number of ALDH bright cells detected within a contaminated background of cord blood cells to be within 20% of the expected values. Protocols for testing each of these parameters are written and submitted for verification by the FDA prior to testing. Accuracy, repeatability, and linearity were tested in the same experiment. A single operator tested segments of known concentrations of ALDH bright as percentage of viable CD45. These segments were created by purifying CD34 and CD133 cells from a cord blood unit using magnetic cell separation. We spiked these cells back into the bulk of the CBU at specific concentrations and froze the blood into segments. The specified range of concentrations for the ALDH bright cells were 0.05%, 0.1%, 0.5%, 1%, and 3%. These were chosen to mimic the range of values that we have obtained on segments from banked cord blood units. The 3 segments were assayed at each concentration for the percentage of ALDH bright cells in the viable CD45 gate. Here, accuracy is defined as the closeness in agreement between the value which is accepted, either as the conventional true value or the accepted reference value, and the value found. The tested samples must therefore have a ALDH bright percentage within 20% of the expected value. As you can see here, each one of them was for each tested concentration. Repeatability is defined as the precision under the same operating conditions over a short interval of time. Here the tested samples had to have a variability of less than 20%. As you can see, the CV for all of them were well below 20%. And linearity, defined as the ability to obtain test results which are directly proportional to the concentrate of the analyte in the sample. The correlation, seen on the right, between the target values and the mean tested values of samples needed to have an R-squared value greater than 0.75%. In this case, our R-squared was 0.98%. So all 3 of those parameters passed. We then set about testing the intermediate precision, defined as the within laboratory variations, that is different days, different analytes, and different equipments. We tested this using 5 cord blood units. These units were processed and divided into multiple segments and cryopreserved. Segments from each CBU were then tested by 2 operators on 2 cytometers over the course of 6 days. So each dot here represents 1 day of the assay over 6 days, whereas the operator 1 on instrument 1, operator 2 on instrument 1, operator 1 on instrument 2, and operator 2 on instrument 4, and then the 5 different cord blood units. The CVs were then calculated for all cord segments within a cord by operator or by cytometer. In all conditions, the CVs were lower than 20%, therefore the intermediate precision passed. For specificity is defined as the ability to assess unequivocally the analyte in the presence of compounds which may be expected to be present. ALDH bright cells typically comprise significantly less than 1% of the total nucleated cells in a typical cord blood unit. The primary impurities of the testing, therefore, are other cell types that are expected to be present, such as red blood cells and monocytes that express low levels of ALDH bright, or ALDH, the enzyme. Specificity was tested by creating segments with 20,000 purified CD34 and CD133 cells spiked into 1 × 10 to the 6, 3 × 10 to the 6, 5 × 10 to the 6, or 10 × 10 to the 6 cord blood cells. The cytometer was then selected to detect 5,000 ALDH bright cells in each population. These samples were then analyzed on our standard flow cytometry software. In each case, we were able to detect accurately the 5,000 expected cells, though at the extreme concentrations there was a great amount of variability. Following the validation studies, we submitted that data to the FDA, and they accepted that this is the validation of our assay. Following the earlier retrospective study, we made the decision that segments from all cord blood units requested for confirmatory typing through the Carolinas Cord Blood Bank would be assayed prospectively. Each segment is assayed for CFUs and the ALDHbr alongside the HLA typing. From February 2010 to February 2017, segments from 5,370 units have been assayed. Though there is variability in the segments, some trends are beginning to emerge. As seen below, we've detected a range of 0 to 2.75% ALDHbr within the viable CD45 population, with a median of about 0.34%. CD45 cells had a median viability of 66.89 with a range of 8.72 to 99.02. Of the CD45 cells, 0.66% co-express CD34 with a range from 0 to 6.86. And finally, CFUs range from 0 to 530 total colonies per 100,000 white blood cells, with a median of 52. In the confirmatory typing assay, viable ALDHbr cells correlated well with the CFUs from the segment with Spearman row of 0.766, seen here. In contrast, the viable CD34 cells in the segment were much slower in comparison to correlations with the total CFUs, with the Spearman row of 0.26. ALDHbr cells correlated with CD34 cells with a Spearman row of 0.49. The differences seen here are likely due to how metabolically active the cells need to be in each measurement. Cells in the CFU must be able to divide and grow, while cells detected by the ALDHbr assay have to have an enzymatic activity, and the CD34 assay is just a cell surface antibody test. These observations are particularly important at the lower range of ALDHbr. Almost all of the core blood units that do not grow CFUs have an ALDHbr percentage below 0.2%, with the vast majority being below 0.1. Because of this observation, we can use the ALDHbr percentage to accurately predict CFU growth. This is important because the ALDHbr percentage is available the day the assay is done, as opposed to the CFU assay, which takes up to 2 weeks to get results back. Therefore, the ALDHbr assay can be used as a prospective assay because the average amount of time it takes from when a cord is selected to when a cord is actually sent to a transplant unit is about 11 days. Interestingly, the correlation seen before between the ALDHbr cells, the CD34 cells, and CFUs seen in the previous slide are reasonably consistent regardless of the amount of time the core blood unit was in storage. That is, the total samples here, less than 5 years in storage, 5 to 10 years in storage, and greater than 10 years in storage. As you can see, in each case, the correlation between the ALDHbr and CFU and CD34 and CFU was consistent over time. This is possibly an indication of stability of the frozen core blood units over extended period times of up to 18 years in this case. Between July 2007 and August 2009, samples from fresh core blood units prior to preservation were assayed for ALDHbr. As CT testing has been done, segments frozen from those CVUs have also been tested through the CT testing. Comparison of data from the fresh core blood units and frozen segments revealed a strong correlation with a Spearman row of 0.43, though segments tended to have a lower ALDHbr content after frozen. In this case, the red line here indicates where perfect equality would theoretically be. We also studied the correlation of the potency assay between cryopreserved segments and the bags themselves. Research units from the Carolinas Core Blood Bank were randomly selected. The entire unit was thawed and assayed, along with the distal segment that would normally be assayed for CT. In this case, the unit was thawed according to the protocols used to thaw units for transplantation. While, once again, there's a strong overall correlation between the segments and the units with a Spearman row of 0.88, segments still tended to underestimate the ALDH content in the unit slightly. We've also asked whether ALDHbr, CD34, and CFU from the segment potency assay correlates with engraftment in a cohort of 78 patients transplanted at Duke with a single core blood unit. 94% of the patients, 73 out of 78 of the patients engrafted. In this case, there were 18 malignant and 55 non-malignant diagnoses. The median time to engraftment was 16 days with a range of 10 to 38 days. The association between the segment-based ALDH-Bright and the CFU and this subset of segments was similar to that observed in the previous set of segments that I showed with a Spearman row of 0.7. We observed a significant trend towards faster engraftment with increased expression of the ALDH-Bright. In this case, segments that from units that had greater than 0.5% ALDH-Bright engrafted significantly faster than segments with a lower ALDH-Bright levels. In this cohort, the CD34 and the CD45 or CFU did not predict faster neutrophil engraftment, though the data is not shown here. Overall, caution should be warranted in interpretation of this result due to this relatively small subgroup sizes, and we're continuing to add data to this as more units are transplanted. Our current research with the potency assay is proceeding along several lines. We've been working to build a database of units that have been transplanted where the CT potency assay has also been performed. The potency assay is being updated to add a red blood cell removal step with magnetic separation. This was a system developed at STEMCELL Technologies called the RethoClear that we've integrated into our assay. We also are preparing to make a switch from out-of-count to out-of-fluor. The out-of-count kit, which uses individual tubes of reagent, is no longer commercially available. The out-of-fluor kit, which is produced by STEMCELL Technologies, is packed as a bulk reagent and will be used in the future. We're also switching over from using separately purchased antibodies to a kit that's available from STEMCELL Technologies that contains all the antibodies and dyes used in the assay. In addition, we are preparing to transfer the potency assay to the CLIA-certified Carolinas cord blood bank, so that the assay will be performed as a regular test of CBUs in the bank and will no longer be strictly research. As I showed earlier, red blood cell content of the segments is high. This can make analysis of the samples difficult. Part of our current research is examining ways to remove red blood cells without contaminating the assays. In conjunction with STEMCELL Technologies, we studied the use of a RethoClear to remove red blood cells. This simple procedure is done following the wash step of the assay prior to the incubation with the fluorescent reagents. Cord blood cells, which have been washed and diluted in dextroamin, have been pelleted. They are then resuspended in an assay buffer with the RethoClear reagent, mixed together, incubated at 1 minute at room temperature. The tube can then be placed into a magnetic field. After 1 minute, the bound cells are moved to the side of the tube, and the unbound fraction can be pipetted directly into the paired out-of-count reagent tube. To show the effects, as I showed earlier, red blood consent, the exclusion gate is set around the similar group of cells in the new assay as compared to the old assay, where here, with no red blood cell removal, you can see that there's a large stained section of cells representing the red blood cells. Following red blood cell removal, there's an obvious reduction in that group of cells, the gliase stained cells. The red blood cell removal has some effect on gating of the CD34 cells and the CD45 cells, but is most obvious in the gating of the ALDH-Bright cells, where you can see the large background of cells without red blood cell removal. Now, with red blood cell removal, shows very obvious groups of cells that makes gating easier. In my standard assay, 100,000 viable CD45 events are collected on the cytometer. The graph on the left shows how many events are required to achieve each level with and without red blood cells present. Without red blood cell removal, the number of events required range from 180,000 to 900,000 with an average of 350,000. With the red blood cell removal, the number of events required range from 150,000 to 200,000 with an average of 170,000, thus cutting the total cells needed to be counted in half, but while achieving similar results. The graph on the right shows the ALDH-Bright content of these same groups of samples, both with and without red blood cell depletion. In this case, the ALDH-Bright number correlates well, though the ALDH-Bright tends to be slightly higher with the red blood cell depletion. As a pilot study for switching from the out-of-count assay to the out-of-fluor assay, 71 segments from cord blood units requested for confirmatory typing were assayed by both methods. The segments were thawed and washed as normal, whereupon half the segment was used for the out-of-count assay. The other half of the sample had the red blood cells removed with erythrochlear, followed by staining with ALDHbr and the new stem cell technologies antibodies. Comparison of the ALDHbr as a percentage of viable CD45 and the CD34 cells as a percentage of viable CD45 were then done. In both cases, there was a good correlation between the assays, though the ALDHbr assay with the red blood cell removal tended to have higher percentages of ALDHbr and CD34. Statistical analysis, however, showed that similar cutoffs could be used for predicting CFU growth in both cases. Therefore, we are moving on to proceed with switching over from the out-count assay to the ALDHbr assay currently. In conclusion, higher ALDHbr from cryopreserved cord blood units predicts faster time to engraftment after umbilical cord blood transplantation. The content of ALDHbr as measured on segments correlates well with CFUs and can be used in a significantly shorter time to predict what the CFU growth will be, and therefore can be used as a potency assay. The assay can be performed on segments so that the cord blood unit does not need to be thawed and compromised. It also allows for these assays to be done at the time of HLA typing without using an extra segment. Assay results are then available in less than a day and can therefore be used to select cord blood units with high potency and to identify cord blood units lacking potency to prevent release of those units from the bank. And finally, we are continuing our prospective studies to validate these findings. As I said, our current goal is to move our assay out of the research space and into the more highly regulated clinical spaces so that we can use this as an actual release test for cord blood banking. I'd like to thank several people who've helped me with these studies, particularly Joanne Kurtzberg, who's my mentor and has assisted me for years in developing these assays, Kristen Page, Andy Balber, and Jesse Troy, who've helped with advice and statistical analysis, Tracy Gentry, who worked at Aldergen and then at Duke, assisting me in performing these assays, and then Barbara Walters-Pick and Dondi Purcell, who work at the Stem Cell Laboratory and the Carolinas Cord Blood Bank, respectively, and have supported me in all these efforts. And with that, I will take questions.
Thank you so much, Kevin. Yes, so that now concludes the presentation segment of our webinar. We will now look at some of the questions that have been submitted. So we have one question. How does the gating, could you elaborate on how the gating works with having both 7-AAD and GlyA in the same channel?
Basically because we are going to be excluding both the dead and the red blood cells, we decided that having 7-AD and GlyA next to each other wouldn't affect the overall assay. In the actual practice, the GlyA and the 7-AD are generally separated so that the GlyA stained red blood cells are actually brighter than the non-viable 7-AD stained cells. If you wanted to, you could probably gate the two groups separately. But because in our assay we're excluding both the non-viable and the red blood cells, we figured there wasn't a problem with just using both in a single gate.
So have there been any inter-laboratory studies to look at reproducibility of the assay when testing the same samples but with different labs?
We've done some of that. The main challenges with testing between different labs is that there really isn't a standard that you can use as far as you can't purchase just ALDHbr cells by themselves and test so that the two labs are separate. So we've tried to test both groups, and we've had some difficulty in that different cytometers, particularly the fax calibers, we had difficulty using, because there were so many red blood cells, the fax calibers had a difficulty counting accurately. Part of our current research is to try to shore up the control samples that we use, because every day when we test our CTs, we run a control from a cord that we've tested multiple times so that we know roughly what the ALDHbr and the CD34 should be in that particular control. And then that way we can verify that each day there's not been an error within the test itself, for instance, not adding activator or something like that. So we have an internal control. When we export to other labs, we are working on getting those controls more standardized so that we can send those controls to another lab and make sure that they can run and get the same results that we get.
There's a technical question here. So preparation of the cells for ALDHbr requires a number of spinning steps. Have you seen any loss of cells or sort of an effect on the recovery of ALDH-Bright cells? And is there a way to perhaps do the test without multiple spinning steps?
We've tested different spin speeds, and what we found is that unless you get very hard spins, we don't tend to lose the ALDH-Bright cells in the spinning. The hardest spin we do is 2000 for 30 minutes in the dextroalbumin because of how viscous it is. And there is some loss of CFUs in particular at that step, which is why we take our CFUs before we spin. In our testing, we didn't find a loss of the ALDH-Bright cells themselves from centrifugation. And we haven't really tried just taking the dextroalbumin and putting that in the reagent itself. We have not actually tried that.
All right. For labs that are currently using the CFU assay as a potency assay, and maybe in the adoption of this new assay, do you think would it be advantageous to transplant using, or make the decision to transplant using results from this assay, but then also set up a CFU assay with the same sample, just sort of for more information later on?
Well, currently we use both assays on all our samples. The ALDH-Bright assay gives a good indication of whether or not the unit is going to grow colony-forming units. However, at the very low end, when you get below about 0.1%, as I said, the samples that grow CFUs or don't are much more variable than the rest of the samples. So about half of the units that have an ALDH-Bright below 0.1% will grow CFUs. So for those units, we like to look at both results before deciding whether or not to use them. But we feel that just the ALDH-Bright by itself, as long as it's above 0.1%, is strong enough to use as a release criteria. But currently, I think using both assays will be advantageous for the near future anyway.
One quick question is where the events come from that are shown in plot C of the slide that you currently have up. So the side scatter versus CD45 plot.
The green dots in that, those are actually the ALDH-Bright cells. That's where they appear on the CD45 plot. So they're actually slightly dimmer than most of the cells, which would be in the bulk about above 10 to the 4th fluorescent units. But the green cells are the ALDH-Bright cells. And we look at it like that just because it makes it easier to see where the gating should be, because that's how they always show up.
So, but this entire plot of C, it's coming from B?
It is the cells that are excluded from B. So it doesn't include the 7AD and the GlyA positive cells. It's everything else.
So is there, I think this may have been mentioned briefly, is there a passing criteria for ALDH percentage? So is there a cutoff that a unit needs to achieve to be considered potent? And if perhaps a sample doesn't pass that, is that a reason to then not transplant?
Yes, like we said, our current cutoff is 0.1%. ALDH-Bright as a percentage of viable CD45. That's the cutoff where we want to look at the CFU assay. Above that, very few above that percentage don't grow CFUs, and therefore currently wouldn't be considered potent. As time goes by, we may start to decide that a unit that grows, for instance, 1 CFU isn't really worth bothering trying to transplant. But currently we just look at growth of CFU versus no growth and use the prediction of ALDH-Bright based on that of 0.1%.
Lots of questions. This is great. For the segment versus the fresh cord experiments, did the thawed segment get tested immediately after thawing, or did the cells get time to recuperate from thawing? So there are some studies that say that it may be important for recovery time post-thaw to regain metabolic function in these cells.
They don't get a recuperation time, except for the fact that it takes a couple hours between the time they're thawed and the time they're actually analyzed, just because they're incubations and centrifugation steps. So it takes about 2.5 hours. But we don't, for instance, thaw them and let them sit for a period of time and then assay them. In the past, when we've done that, we've gone up to an hour with them just sitting in the dextran albumin, and for that amount, for an hour, it hasn't made a difference. So I don't know exactly how long you could let them sit there, but in our studies, it hasn't made a difference.
Great. And one last question. Is this something that AABB and FACT should look at having implemented as a standard for better hematopoietic stem cell transplant outcomes?
I think they're looking at lots of different assays. They certainly are interested in this. So we are trying to get together the data to really support using this as a potency assay. The problem is in transplants not done at Duke, it sometimes takes an extended period of time before we have data that we can then subsequently analyze. And we've tried to only analyze single cord blood units, whereas if there's a double cord blood transplantation, it can be difficult to tell whether or not our unit was by itself, or just to do the statistical analysis when we only have one of the units tested can be difficult. So we're trying to get that data together, and certainly we are discussing with FACT and AABB that the use of this assay, but I don't know that they are quite ready to use it yet.
Great. All right. Well, that concludes the Q&A session of our webinar. Thank you so much, Kevin, and thank you to everyone for joining us this morning for this great presentation.